And so first and foremost, I want to invite everybody who are friends of the chemistry department to the chemistry department picnic, which is next Wednesday at 4:30 in City Park. You do need to sign up outside of the stockroom. For those of you that don't know what a stockroom is, just go down by the elevator and there's a sign-up sheet. Daniel over here can make sure you know where to sign up. So I want to point that out. Number two is that after Shelby's talk, 5:10, we will be having Emily's talk. And so you are also invited to stay for that particular talk. Next week we are going to have yet 4 more talks. This is going to be, what, this is talk, what is it, 14 out of 18? And so you're invited to come to those. If you are big into binge watching, okay, so you want to sandwich something in between Friends and Stranger Things, this is your opportunity. There are 77 of these up from the last 4 years. You can watch those on YouTube by just Googling Lawrence Chemistry and then Seminar. And so you can watch all of these so in perpetuity. So it's my great pleasure to introduce Shelby. And so it's really, I don't know, it's kind of that thing, you get a little verklempt, so to speak, from a Saturday Night Live sort of thing, looking over and having research students and remembering how did they wind up in lab, okay? And I was trying to figure out how Shelby wound up in my lab working for me. And so the very first time I remember just seeing Shelby, she came into the biochem lecture hall. She sat down like right in front, okay? And I noticed only because the guide dog was sitting right there. It's like, oh, somebody knows the guide dog. And that she had the classic resting bitch face, which I didn't really realize at the time. It was just that, well, you guys are very familiar with it. It's just that, and I knew I was in trouble when I cracked the first couple of jokes, and there wasn't a smile. And it's like, okay, there are problems here. I later learned out that she was really laughing inside, which is good. We then fast forward to in lab, and I tend to block out lots of different things, but I do remember talking to her and her lab mate Rachel and going, you did what? And then just kind of nodding my head and then just walking away and going, oh my, okay. And it was really interesting because that was the Western plot, which she says was all my fault, but I don't think that was necessarily the case. Anyhow, what then subsequently happened is that she came to my office, and I didn't realize until later that took a lot of courage to come in and go, hey, I'd like to do research with you. And that I always am suspicious just a little bit of somebody that sits down and like flashes a smile, and you're like, am I being bullshit at this particular point or not? But we started working on a virus project together. And I found at this particular point that she had this love of bodily fluids. She totally took into like running around, about a bunch of you around here gave a nasal lavage, okay? Somebody ran up, oh, give me nasal lavage. And so that I then realized at that particular point she had a talent and started to love doing research. I also realized at this particular point that she was quite nervous to be doing these sort of things. I suffered the same thing when I had my bosses come by, as my hands would start to shake, and I would come over and her hand would start to shake. And that and that I start this particular way because Shelby has grown tremendously over the last 2 years doing research with me. And it really makes me very proud to see how she's developed as a scientist. That first bit of uncertainty inviting her into the lab, as I had mentioned with Michael's introduction and with Lindsay's introduction, each person that joins the lab adds a little something special to it. And Shelby brought with her that empathy. So that there was always, she was always going to be the mom of the lab, always the cheerleader, okay? And that was really nice. I mean, and I was a little bit surprised by this because she also does this for like the swim team. So it's like I come in and I go, did you look at the cells? And she goes, no, I was at the emergency room. It's like, weren't you there like 3 days ago? She goes, yeah. It was like in like the first month, like half the swim team she took to the emergency room or something like that. It was only a quarter. It something like that. But that really, meant a lot. And that, we also started to figure out with the bodily fluid thing that she had a proclivity and loved blood. Okay? Not only, and keep in mind, a lot of students like to work for me because they get a chance, if they want to draw blood, to stick me. Okay? And most of them are pretty good at it because they've already had a little voodoo doll in Biochem 1 and they poke it. So they're usually pretty accurate. By that same token, I discovered that she likes being stabbed as well, and you're just like, oh my gosh, because I would get like a little email that's like, look, I'm donating blood, and I'm like, no, we need that for the research. Yeah, we can't bleed you for 2 more weeks. No, we can't do that. And then, of course, you guys are all very familiar with Shelby's sarcasm. And so back and forth, actually, she wound up with all kinds of different nicknames, like Scruffy, because she'd come from the pool and directly in and start working, late in the evening on various things. Or Cinderella, because I'm kind of the messy person in the lab, and she's like, oh, for the love of God, don't do this way. But I'm really proud that I wound up having her do this particular project. And so this particular project that you're going to hear about was one of the very first ones I brought to Lawrence. And that for a variety of different reasons, this particular project is winding down, and that there are a lot of very valuable reagents, and so that it took a lot of it, for me, it's a very kind of emotional project, and developed trust between the two of us to work on this particular project and get it to work really well. It's just been a real pleasure to have her. And I've never, really something else is all I can say in terms of getting this sort of data put together. So it's going to be an excellent presentation, and here I give you Shelby.
Thank you all for being here. It means so much to me. Not going to cry till the end. But thank you, especially on this beautiful day. I appreciate you coming indoors and sitting in the halfway dark room. So let us begin. So I am doing a presentation on my research that I've done with Dr. Dave on gamma-aminobutyric acid and its ability to mediate interferon beta in the presence of human rhinovirus. And that sounds like a mouthful. And you're like, what's going on? But never to fear, your tour guide is here. So here, let us begin. So raise your hand if you know of or are a person that suffers from asthma. So if you look around the room, that is so many people. And asthma has been on the rise, globally and in our own, on our own front porch. Exactly 25 million people in the United States in 2010 were diagnosed with asthma, and that is continuously growing. And with that, you have to think about lost work days, lost school days, and all the medical bills associated with this. And that cost the healthcare system approximately $56 billion with a B dollars. And that's very significant. And also, there is unfortunate consequences of asthma too, and asthma attacks. Approximately every year, 3,615 people die from asthma exacerbations or asthma attacks. And 219 of those each year are children under the age of 18. So even though we joke around sometimes with people that have asthma, are like, oh, I need my puffer and all that stuff, it's actually more serious than people lead on to. So what about asthma? What is it? What is the physiology of it? So essentially with asthma, they're chronically inflamed airways, and something triggers this. And when that happens, there's a smooth muscle contraction in your bronchioles, or your airways. And it will cause the smooth muscles to contract around, that's the tightness in the chest. Simultaneously, there will be chronic inflammation and mucus secretion. So this lava-looking stuff is mucus. So we all have mucus in our lungs, and it's at normal levels. So it will expel the dust, pollen, whatever we get in our lungs normally. However, someone with asthma, this is increased tenfold. And so both of these together cause the symptoms of asthma. So this is the chest, elephant on my chest, tightness, the inflammation, and the increased mucus is the coughing and the iconic like wheeze. That is that. So the muscle contraction, though, is very important. But it's the inflammation that does the most damage and people don't really know as much about. So what causes this inflammation? What is the inflammation? And it's due to an inflammatory response. So again, interactive. Raise your hand if you have ever been sick. Raise your hand if you have suffered a horrible, just truly excruciating paper cut. Like I know, there are tears. So this has inflammation within it. And inflammation is essentially the pain, redness, and swelling that is involved in any type of infection or damage to your skin, your body, sometimes mentally, But essentially, so let's have a little scenario. So you're a bright-eyed and bushy-tailed Lawrence student. You're walking up the steps of Wards. You're really excited. It's a great day. And then you trip and bash your knee on the Wards steps. And you're crying. And you're contemplating leaving because it's just what else could go wrong on finals week. And you pull off to the side. And once the blood and the tears stop, then you see that your knee is hot and it's red and it's swollen. And so what is it? Why is this occurring? Well, it's really important because it's a defense response of the body. And so what happens are these small little molecular patterns will be sent out, and there's ones that are specific to pathogens and ones that are specific to damage. And essentially what it will do, well, the first step, it will cause vasodilation of the capillaries. So essentially it will cause an expansion. So this allows more fluid, increased fluid, so the swelling, and that also increases the immune cells to come in and fix the problem. So you need those. So with all that in mind, this is what occurs with the inflammatory part of the disease. But what essentially triggers it? Well, there's, just like people with allergies, there are lots of different stimuli that can trigger. So you can have, Cujo and other furry friends. You can have pollution from the nice smokestacks over by the paper mill. You can have dust from on top of your textbooks because you never open those. You have chemicals from the Orgo lab. You've got mold from the microbiome lab. And then you have infections. And it's these infections that are what truly are the interesting part of asthma, virally induced asthma exacerbations. And that's what Dr. Dave and I were focusing on. And so with that in mind, you have to get into the mindset of a virus. So I'm going to get a little interactive here. So it's going to be like Oprah. So, all right, who's ready? You're going to get a virus, and you're going to get a virus, Meg. Come on, there you go. Everyone getting a virus. Oh, way in the back. Virus. Who's getting a virus? Oh, you're getting a virus. Sam, you're getting a virus. All right, swimmers are getting a virus. Oh, there you go. Still have more. Virus. Woo, nice. Okay, virus. And Matthew, get a virus. Oh! Wow. Okay, that's aggressive. But anyway, so we have to get a little infectious. So, unlike Oprah, I'm giving out viruses. And the reason being is to make a point understood that viruses infect us all. And specifically, we are looking, Dr. Dave and I were looking at a specific family of viruses, which we'll go more in depth, but specifically the picornavirus family. And if you aren't familiar, here's some of the fun relatives that are associated with picornavirus: foot and mouth disease, polio, and hepatitis A. So it's a good time with this family. So there are over approximately 80 different species within the family in 35 genuses. And all of these viruses are small, non-enveloped, or slimy, as a virologist would say, single-stranded RNA viruses. So the RNA portion is what is being transferred within the virus. This is the precious material. And there's one that we left off here that's very important. Some of you have caught it in the audience. There's a green one and two purple ones. So they're very small, and they are the deadly. Just bear with me. They're very deadly. It's scary. Human rhinovirus. I know. Calm down. It's okay. It's okay. So human rhinovirus is the most common viral agent in humans. So think of that. Out of all the infections, let's say in a year, most of them are from human rhinovirus. That's kind of crazy. I don't know about you, that's kind of crazy to me. Human rhinovirus is transmitted from person to person via contact, physical contact, or aerosolized. So those barbarians that sneeze during cold and flu season and they don't cover their mouths? Yeah. Thanks to them, a lot of us get infected. It targets epithelial cell linings, and guess what your nose and your throat and your airways are made out of? Epithelial cells. And it's responsible for the common cold and asthma exacerbations. And you're thinking, well, asthma exacerbations? Asthma attacks being caused by this. Well, yes, that's approximately 50% of all asthma exacerbations are caused by the human rhinovirus, which I did not know previously. So if you look over here, this is an intact epithelial cell lining. This is a normal lung. And here's the human rhinovirus coming in. The epithelial cell lining, when it is healthy and not damaged, it will catch the virus and it will prevent, usually most of the time, from infection. However, this is a schematic of someone with chronic inflammation like asthma. And so there is so much damage being done by triggers, and these inflammatory cells keep popping up because they're being responsive to the triggers, and the inflammation causes more and more damage, and this allows the little viruses to sneak on in and infect. So with this all being said, these viruses are really a pain in the butt for the epithelial cell lining and someone with asthma, but it's the inflammation that is causing this degradation. So you need a little bit more information on the immune system, because not all of us, unfortunately, have taken Dr. Judith Humphrey's immunology class. It's a really good one. I recommend it for those of you staying here. So we're going to have to learn about the immune system. Well, we learned in 10 weeks in less than 5 minutes, so bear with me. So there are two parts of the immune system, the adaptive immune system and the innate immune system. And the adaptive immune system is affects you days after post-infection, and it's very specific, it's very diverse, it's very targeted. And an example is vaccines. You know, you don't want polio, so you get a vaccine and it won't work right away. So if someone, you know, is walking around with polio and you just get the shot, like, that does not mean you're going to get covered there. So this is the basis behind vaccines. You obtain memory. The innate immune system, on the other hand, is activated immediately. So once you get infected, these molecular patterns are going to be sent out, and that's going to be like a flare gun or the bat signal, and they're going to be running that way, the immune cells. And they're very general response and same every time, because they're patrolling constantly. You know, they cannot be very diverse and specific. So the way to parallel this is the adaptive immune system is like the Navy SEALs. Very specific, they got their target, they're out to kill. The innate immune system, on the other hand, still out to kill, but you know, they don't have the equipment that these guys do. They're foot soldiers, they're the infantry. Very important still, but not as specific. So the adaptive immune system consists of T cells, B cells, and antibodies. And the innate immune system consists of physical barriers like your skin, and phagocytes, which are eating cells. And specifically the immune cell from the innate immune system that Dr. Dave and I have been focusing on is this really nifty little guy called macrophages. And they have an amazing ability they are part of the innate immune system, but they're able to communicate with the adaptive. And essentially the way to parallel this is the macrophage, let's say, is a cat. So your cat catches a nice fat mouse and walks up to your bed, drops it on your pillow. So you know you have a rodent problem. And so you can dispose of this efficiently, and you can deal with the problem efficiently. But if you didn't have that cat, you wouldn't know you have a rodent problem, and that would cause a lot of other issues. So that's why they're important. But essentially, we'll go in depth about why they're so important. So here's a fun little schematic that I like. Isn't he so cute? Anyway, this is what they really look like. Not that cute, but oh well. So they have a lot of strengths, and one is their large appetite. They're essentially the Pac-Mans of the innate immune system. Macrophage is Greek for to devour the cell. So it's very, very crucial in the innate immune system for getting those pathogens. At the same time, they're very fine-tuned at sniffing out these pathogens. So immediately when you're infected, they know, and they're going to be out to kill. However, let's say like you have salmonella, and you got a lot going on there, a lot of replication of the bacteria, and they can't handle it all by themselves. Well, this is where they are able to talk to the adaptive immune system and say, hey, we need help, we need backup. And how they do this is quite interesting. It's a game of balance and telephone, I would say, but not like screwing it up purposely telephone like we all do. It's not that. It's very efficient. They do this by these very, very small molecules called cytokines. And essentially they come in two different flavors. They have anti-inflammatory and pro-inflammatory. So an example of anti-inflammatory is IL-10. And an example of pro-inflammatory is TNF-alpha. TNF-alpha is what makes you feel lethargic and loss of appetite when you're sick. IL-1 beta is what racks up a fever. So these are pro-inflammatory. And the thing with cytokines is you need a balance. Balance is key. It's like yin and yang. You need it to be even. You want the level, you want the scale to be even. You don't want one more than the other because it could cause severe problems on either side. However, people who have asthma and they are infected with viruses, with virally induced asthma exacerbations, it skews the pro-inflammatory cytokine all the way to the left, or to the right, I should say. And it skews it in the way of the pro-inflammatory. And this will cause significant amounts of damage. So we need to keep this in check. So there has been work, my predecessors in Dr. Hall's lab, that worked on approximately, I don't know, between 4 and 8 years ago, but on the specific key inflammatory cytokines, these pro-inflammatory cytokines. And there is 2 papers. And one was essentially finding out the pro-inflammatory cytokines that are important that macrophages express when they are presented with a virus. So this is specifically viruses and what macrophages express with viruses. The second paper is about activation of different pathways of macrophages and asthma-relevant cytokines. So these are specific to the inflammatory disease. And then both had a little bit of mix, but that all these mechanisms behind these inflammatory mediators were virus-specific. And that makes sense because one shoe doesn't fit all. You know, one size does not fit all. They're going to be different for different viruses and different, you know, it's not the same. It's going to be a little different. So with that all in mind, the question was, how can we attenuate the immune response to prevent chronic inflammation? Because this is important. There's lots of chronic inflammatory diseases out there. So starting with asthma, we need to figure this out. So you can do this by a nifty little neurotransmitter called GABA, or gamma-aminobutyric acid. I know, calm down guys, it's really exciting. I know you guys seem so excited about it, but I'll tell you why it's very exciting. So this is what GABA looks like. So all the chemists in the room, didn't do this on ChemDraw, so sorry. So essentially what it is it's this really cool little molecule, and it has this really cool ability to inhibit. So it essentially is able to pump the brakes, or essentially stop sometimes what is going on. And so it is primarily found in the central nervous system, and that was originally the idea, that it was only found in the central nervous system. However, there are whispers, rumors flying around. You know, science gets crazy out there. But that it might be in the immune system, these receptors that would have GABA bind. And so it wasn't really sure, and it was found in the adaptive immune cell, because T cells are in the adaptive immune system. So the question that kind of came about was, if GABA is in the immune system, could it possibly be in the innate immune system? Because there's whisperings of the adaptive, could it be in the innate, and could we use it to attenuate these macrophages and essentially inhibit or mediate some inflammatory properties? So before that, you need to know a little bit more about GABA. I know some of you are dominantly chemists, so we're going to work on that. So it's found primarily in the central nervous system and in the immune system, fun fact. And so you need to know a little bit about cell biology. So you're going to have your lipid bilayer of cells, inside a cell, outside a cell. You're going to have your GABA-A receptors. You're going to have GABA, and you're going to have some chloride ions. And essentially what will happen is GABA will bind to the receptor of the GABA-A, and it will cause this chloride ion channel to open. And this will cause an influx of chloride ions. And essentially this makes the inside of the cell more negative than the outside. And this is called hyperpolarization. So it essentially is pumping the brakes. This leads to its significant inhibitory properties. It's not able to send signals. It's able to, whatever the cell is doing, it will pump the brakes on. And so with all of this being said, what was previously found about GABA? So this is where my research came to be, was from my predecessor, Bethany Condell. So Bethany was very interested in seeing if GABA receptors were in the innate immune system on macrophages. She was very interested in this. So she did research with Dr. Dave, and they discovered that there were GABA receptors found in the immune system, and specifically on macrophages, which means in the innate immune system. So that's super cool. And they also found that GABA attenuates inflammation in macrophages exposed to bacteria stimuli. So that's kind of a mouthful, so I'll just show you what the results mean. So this is the results, and for those of you who don't read, fun bar graphs all the time, you have picogram per milliliter, which is essentially the amount of TNF-alpha, that's that lethargic, loss of appetite cytokine. That's essentially how much is being secreted. And these are the test subjects. So you have control, which is just macrophages in the well. You have GABA, so macrophages plus GABA in the well. Macrophages plus a virus in the well. And macrophages plus GABA plus a virus. Not a lot happening, not a lot going on here. However, if you look to your right at these beautiful, very extreme bar graphs over here, this is where the cool stuff is happening. So lipid polysaccharides, so this is called, yes, LPS, lipid polysaccharides, and they are found, if you know anything about bacteria, gram-negative bacteria, this is what the outside of the cell consists of. So if you drop a little piece of LPS in a well with macrophages, they will go crazy. They will be like, kill, 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 I'm out to get him. So, of course, that makes sense that macrophages, if you put LPS in there, would cause an influx of TNF-alpha. But what was really neat is when you put in GABA, it's this drop that is statistically significant that is so freaking cool, because GABA will cause this drop. It will pump the brakes on this inflammatory cytokine when you put it in there. That's really neat. If you know anything about medicine, this is very applicable if you can pump the brakes on a chronic inflammatory disease. So that's awesome. So with all this in mind, based off of Bethany's work, is GABA important in viral inflammation? And that's kind of where we came into it. Would GABA, if you put it in with macrophages, what would it do, and could it affect the antiviral response? That's where the research question came in. What was it, a year ago? Kind of crazy and insane, but this is where it all began. So will GABA serve as an inflammatory mediator and alter cytokines that are released upon exposure to human rhinovirus? So that's our idea. We're doing more of the virally induced asthma exacerbation focus. So you need a game plan, because when I first came in, as Dave said, I was nervous, I didn't know what the hell I was doing, I was getting scared. So we had a game plan, we found this all out, and once you repeat this over and over and over, you get used to it. So you have to bleed people, because contrary to popular belief, you can't just go, get macrophages off of trees. You can't wipe this and put it in a well and be like, macrophages. No, they're in your body. And so you need to essentially purify blood. And from that, you will plate them in 12-well plates and do time trials. So test out GABA and the viruses and the antagonists and see what's all going on in there. Then what you're going to do is you're going to isolate the proteins and the cytokines, and then you're going to analyze. So let's begin. So you got to do some phlebotomy. Didn't know about that beforehand, but what you're going to need is you're going to need your utensils. Okay, it's very important. So you're going to need some, alcohol wipes, sanitation. You don't want anyone getting sick. You need a tourniquet. So you're going to put this up here, and you're going to get ready. Think of happy thoughts. You're also going to need a 21 gauge butterfly needle, all single use. You're going to need 2 of these. These are 60 mil syringes, and you're going to need a 16 gauge needle. And very scary. But you're going to hook that up, and you're going to swab so it's intact and beautiful. And you're going to try to hit that median cuboidal pipeline right here, this vein. And that's usually where you're going to get the most results. So I'm not going to do it. I know you guys thought I wasn't. So, but you're going to go in, not with this one, but with a butterfly, and you're going to go at a 45-degree angle. And once you get, you tap the pipeline, as we would say, you are going to drop that blood. And essentially, we will be taking approximately 120 milliliters of blood, which is 1/5 of a donation. So after you do that, you got to do blood density separation, because you can't just throw the blood in a plate and go, oh, we're done. No, that's not how that works. So you're going to have to get some gloves, you're going to have to get some goggles, you're going to have to be Vogue-esque for your picture, and you're going to have to start pipetting. So you're going to pipette the blood into conical tubes that have some lymphocyte media separation at the bottom. And essentially this will cause density layers. But you have to centrifuge these samples, 2,000 RPM for approximately 30 minutes. And you're going to get this beautiful, beautiful picture. So I'll walk you through it. Down here is your thrombocytes, or your red blood cells. They are the lowest on the totem pole here, but that's because they have iron in them. If you know anything about hemoglobin, they're going to be down here. Up top is platelets, clotting factors, and other, the fluid that is around in your blood. This is going to be up top. This is the holy grail. This is what we're looking for. And this is called the buffy layer, or the buffy coat. And that's where your white blood cells, or your immune cells, your macrophages will all be. So this is the money spot. And that's what you want to get. So you're going to pipette that out, and you're going to use a centrifuge and distill it down some more. And you're going to plate them in some young 12-volt plates, labeled accordingly. And you're going to pipette 1 ml of cells with media, because they need something to snack on, something to survive. And you're going to incubate for several days. You don't want to put in cells right away, because they're going to be freaking out, and you don't want to test them right away, because new environment. And so you're going to allow them to incubate, and as you saw before, macrophages kind of spread out. That's what you want. And once you do that, that's when you can start experimenting and do the time trials. So there's 2 different roads of experimentation. And one, after you plate the cells, one would be a 60-minute time trial. And with that, you're seeing the difference in phosphorylation. And specifically with that, we will talk about later and in more in depth, but you're going to see different expression of proteins, essentially. And so you're going to isolate and identify these proteins. And you're going to do that by SDS page and Western blots. We'll get there, don't worry. And after the other option, the other fork in the road, would be the 24-hour time trials. So you essentially put in virus, and you put in GABA, and you give them 24 hours. That's going to allow them to secrete those cytokines we've been talking about. It's going to be able to secrete them into the media. So then you'll be able to harvest the media and isolate the secreted cytokines and mediators, and then you can test that with ELISAs. So we're going to start with ELISAs first. And ELISA stands for enzyme-linked immunosorbent assay. And this kind of looks confusing. We're going to go by step by step. So you're going to have these 96 well plates. Yes, 96 wells. And you're going to start with coating the cell, or coating the plate. And what you're going to do with that is a capture antibody. So as we talked about before, antibodies are very specific. And this is what is going to, your sample's going to come in and bind to. This is what it's going to be. You can buy these. It's very easy. Well, not really that easy. DMARC, it's not that easy. I'm sorry. So then what you're going to do is, once you tap that all out, you're going to tap, tap, tap it out, and then fill it up with a blocking solution. You're going to block it. So all this space that's not being taken up by your antibody, we don't want anything binding to that. So that's what the blocking is for. So then once you get the blocking done, you're going to add your sample protein, or the cytokines that we were talking about. And that's what this green ball is. That's going to be your cytokine. That's going to be your sample. And that is specific for this antibody. Then you're going to tap, tap, tap it out again, and you're going to add the detection antibody. And this is essentially a flag that's like, I found it! So very important, especially for analyzing and reading out. So that's going to essentially lead to readout and analysis. And it's going to be a color change, which is super cool to see. And I'll give you a little example. These are some really good hombres, not bad. And so, as you can see, there's essentially, it's almost like a gradient. But the darker the well, the more cytokine or protein that is in the well. And essentially, this is the color change from blue to yellow, because we want to stop them. We don't want them to keep producing cytokines. So, what are the cytokines we were focusing on? IL-15, which is increases trafficking of immune cells, promotes elimination of infected cells, and alters transcription. Then we have interferon beta, of course, you know, and antiviral properties, which is very important, and reduces antigen presentation, reduces trafficking of immune cells, and it alters transcription. So the main point of this is you're like, what the hell is going on? Well, IL-15 is pro-inflammatory properties. It increases, promotes. That's pro-inflammatory. That's essential to the inflammation portion of asthma. Whereas interferon beta reduces. That's anti-inflammatory. This is what we don't want. We want this, but we don't. We don't want. Because that means that you're infected with the virus. So that's what we don't want. But that's why we're focusing on it. So the asthma and the virus, 2 and 2. So let's look at the results. I know, this is what you've all been waiting for here. So picogram per mil, again, of IL-15 expressed, and these samples. So, as you can see, not statistically significant. It's pretty universal across the board. There's a little bump But as you can see on this y-axis, it's not like it's jumping from 5 to 500. It's like 5 to 7. So it's not much of a difference. So, you know, wah, wah, that's like kind of sad. So, but this next slide is what's going to blow your mind. Blew my mind. So interferon beta, again, amount of interferon beta expressed and secreted. Controls just macrophages. And this is GABA plus the macrophages. And I don't know if you can see this here. I put it on a dark background so you can see. Look at that. Look at how big that is. That's so cool because GABA essentially promotes interferon beta, and that's antiviral, what we were talking about. So when you get infected, this is what is produced. So that's super cool that GABA will promote that. Like, really cool. Like, it's really cool. So this is macrophages plus a virus. And obviously, a virus is not going to make an antiviral. That doesn't make sense. So, but what is very cool here is that if you put GABA, prime these macrophages with GABA, and then you put in a virus, compare this to this, it's going to still increase in the presence of a virus. It's going to increase this interferon beta, this antiviral, in the presence of a virus to rid the body of a virus. That is very cool. And the implications of that are even more mind-boggling. Because just think, I borrowed this from my roommate. She has asthma. Doesn't have anything in it, so sorry. Can't take it left. So usually there's an albuterol, like a steroid in here, that will relax the smooth muscles. That's usually what it has an asthma inhaler is in it. Just think though, if you had an inhaler that had this this steroid that relaxed the smooth muscles, but then it also contained GABA for the cold and flu, well, not flu, but cold season. So then people could be protected by GABA because the inflammation wouldn't be as high. So they wouldn't have as many virally induced asthma exacerbations, which make up 50% of all asthma attacks. That is pretty significant. Like, that's really cool. So I'm working on that patent, you know, just in my free time. So anyway, so how does GABA affect interferon beta expression? Because, you know, chemists, I don't know if you know, they really like mechanisms, so we're going to work on that, okay? So you're going to need to know a little bit about signal transduction. And signal transduction is not very easy to wrap your head around. Some of you that do know, it's not as straightforward as you would like it. So essentially what it is, it's a physical or chemical signal that is transmitted from the outside of the cell to the inside of the cell. And this is done by signaling cascades. So one thing leads to the next thing, leads to the next thing, which eventually alters something. And the end goal is usually altering gene expression. That's the important part. The gene expression will regulate different things such as production of interferon beta. So very important. So we're going to go through this very simply. So this is the inside of a cell. It's a macrophage. This is the outside of the cell. So you're going to have human rhinovirus, and it's going to hang out, and it's going to be like, hey, I need a bind. Because if any of you know about virology, I know some of you took that class. I saw you. It's going to bind here, and it's going to try to gain access, because viruses need cells to survive. They need to get in and replicate and then bust out and do it again. That's how they survive. So they're going to bind here. Well, obviously the macrophage does not want that, so it's going to initiate a cascade. So this is going to change. It's going to have a conformational change. Anyone that's taken a Dave class, size, shape, and charge, that's going to be changing. And this is going to initiate the first cascade, which is where G proteins will bind to this changed receptor on the inside of the cell. Then this is going to be phosphorylated, and this will cause intracellular enzymes to migrate over here, and the G proteins will bind. So again, this is a handoff. The best, easiest way I can describe this is, anyone seen track relays, 4x4? Okay, well anyway, if you haven't, they're going to be running, and then they're going to hand off the baton. That's what it is. And that baton is phosphorylation. So it's going to activate. So again, like if you're handing them off, they're not going to be sitting here being like, oh, I'm going to take that. No, they're going to be running, they're going to be activated, and they're going to take off. So that's what it is. So this is going to cause another conformational change, and then enzyme substrates will be broken off from the intracellular enzymes. They will be phosphorylated, and they will bind to transcription factors. And if you know anything about transcription factors, they are what actually regulate the gene expression. So they will bind, cause a phosphorylation, they will be activated, and they will bind directly to the DNA or a complex on the DNA. And essentially this will say, this whole thing will say from the receptor, hey, we've got a problem, there's a virus, all the way through to the DNA and be like, hey, we need to make interferon beta. So they're gonna pump this bad boy out like none other. So, with all this being said, how do we measure signal transduction? And this is an actual signal transduction, like map schematic. So I simplified it a lot, so I could have done this. Just put that out. I could have done that. I was nice. I was nice. I didn't understand all that anyway. What we're going to do is, this is the fork in the road. This is the 60-minute time trials. And so what you're going to do is you're going to take these 12-well plates, and you're going to have your cells in there, and you're going to prime them with approximately 10 micromolar of GABA, human rhinovirus, or both. And then you're going to give them allotted time intervals. And it's going to be 15 minutes, 30 minutes, or 60 minutes. And this allows for different amounts of phosphorylation. So essentially that handoff. There's going to be different, 15 minutes is not a lot of time for this handoff, in cellular, obviously not actually tracked, but it's going to be not a lot of time. So you're going to see different changes. And then once you stop them, you're going to stop them by lysing them, so they're going to be dead, because you don't want them to continue to phosphorylate. And you're going to do this through this Lamley buffer, and it's blue, so that's kind of neat. And you're going to take these solutions of cells and different phosphorylation, and you're going to do an SDS page gel. Well, SDS page, because gel's in the name. Sodium potassium sulfate polyacrylamide gel electrophoresis. I know, real fun. And you're going to take your pipette and you're going to pipette into each well. So this is a gel right here, and the solution that is in there is sodium potassium sulfate plus some water, and that's essentially a detergent. And the reason why this is important, once you hook this up to a DC current, and you set it for approximately an hour and 30 minutes at 100 volts, you're going to wait, and you're going to think about the inner workings of SDS page while you're waiting. And so it separates proteins by size. And so if you know anything about proteins, they're usually balled up. Well, this detergent flattens them out, and it exposes the hydrophobic areas so that the detergent essentially will bind and flatten them out, so then they can run linearly through the gel, or kind of linear. But anyway, so the bigger the cut of protein, it's not going to go as far. But the smaller the cut, the farther it will go through the gel. And this is the way I can parallel this is it's like a kid in a crowd. The kid can run through legs, dodge everything, and they'll end up on the other side of like Times Square. Whereas we're like, oh, excuse me, like, oh, we're not going to get too far. So once this is all done, you're still not done yet, and you have to transfer it, and you have to live the Western life. And that involves doing Western blots. So you're going to transfer this gel, what is on this gel, to a membrane. And the membrane, the reason we use this is gels aren't really efficient. They will dry out, they'll break in your hand, and then you'll cry because you'll have to do it again. And that will just be a whole segue of crying and sad about things. So then once you transfer it to a membrane, you're going to incorporate a primary and secondary antibody. And then from that, you will image it. And this is what it usually looks like, if you're doing it well. And if you cross your fingers and pray to the lab gods. So, with all that in mind, how does Western blot work? So essentially, you're going to have your membrane protein, and your protein's going to be on there. That's your target protein. And then you're going to put in, with some milk actually, a primary antibody, which is specific for your protein. And then you're going to have an enzyme conjugated secondary antibody. It's very similar to the ELISA, as this is the flag, this is the detection one. So when it is exposed to enzyme substrates, there will be a detection signal, which will be fluorescent. And that's what we can image on blots. So, with all this in mind, we wanted to focus on, this is the schematic I had before, and we wanted to focus on specific sections of the cascade. So the first section, JUNC and ERK, are the enzyme substrates right here, which will directly affect transcription factors, which directly affects gene expression. So that's what we're looking at right here, this part of the cascade. So these are the results. So this is a loading control, so this isn't very important. This is a loading controls. And so we have just a control here, and this is JUNC. And these are the 15, 30, and 60 minutes of each. And so it's GABA plus the virus and just GABA alone. So, as you can see, GABA has no effect on junk phosphorylation in the presence of human rhinovirus. So this is pretty even. There's not like changes in the darker the band, the more phosphorylation, the less dark, the less phosphorylation. So this is pretty even across. However, if you see, GABA alone affects phosphorylation in junk right here on that 15-minute mark. And this is very exciting because this correlates very well with Bethany's, I know way back in this presentation, Bethany Condile's research on lipid polysaccharides, and that it affects, GABA affects the expression of junk, which is really cool to the cascade. This is really neat. So essentially it's reaffirming what she had found out. Then you have ERK, which is another one, again, loading control. And this one is very interesting because, as you can see, you know, GABA stimulates the phosphorylation of ERK by itself, but GABA in the presence of human rhinovirus prevents junk phosphorylation at all time points, which, ooh, spooky. So, stay with me all. I know, it's getting kind of crazy here, but we're gonna find out some really cool things. So then we looked at transcription factors, so this part of the cascade. So IRF3 and STING, which directly bind to the DNA or a complex on the DNA, which directly alters gene expression. So with that, here we go again. We're going to look at IRF3. And IRF3, again, control 15, 30, 60. And this is GABA, human rhinovirus, and GABA plus human rhinovirus. So as you can see, GABA stimulates the phosphorylation in both the presence and the absence of human rhinovirus. So right here and right there. So sorry, that's swinging around here. And whereas IRF3 phosphorylation declines over time in the presence of human rhinovirus. So this is really exciting though because it essentially reaffirms my work that I've been doing that interferon beta is affected by the IRF3 phosphorylation over time. So this increases, this decreases. Pretty cool. I mean, I think. So, and then we have STING, which is another possible transcriptor. This one is more vague. We are still looking into STING, but this is one of our blots just recently. Not as pretty, sorry. And so GABA, human rhinovirus, and GABA plus human rhinovirus. And GABA by itself increases STING. This is a pretty dark band, it's just not as visible. And at 60 minutes, and human rhinovirus at 60 minutes has decreased phosphorylation. So we're still working on the mechanism of this, but this is pretty cool results. So this is the hypothetical model that we have thus far. So we have human rhinovirus and it will bind to a receptor, which will directly, through cascades, affect a transcription factor like IRF3. That will directly bind to the DNA or a complex on the DNA, and that will directly alter interferon beta production. We also have GABA though, because GABA, as we know, affects all of this. So GABA, when it phosphorylates junk, we learned that unfortunately nothing's phosphorylated. It inhibits. So that's interesting. And then, but if GABA is GABA binds to GABA A receptors and it phosphorylates ERK, ERK directly as an enzyme substrate will bind to the transcription factor like IRF3, which will directly bind to the DNA and affect interferon beta production, which very cool research going on here. And that's what we have thus far. So, future work for the paper. What we're going to look at is finish looking at the phosphorylation status of IRF3 and its cousin, IRF7. We are going to look and see if an inhibitor of ERK will prevent the stimulation of interferon beta and IRF3 phosphorylation, just looking into things. And then we're going to find out a little bit more about STING and how this affects the whole model and all the segues and whatnot and the cascades. So, with all this in mind, I'd like to thank a lot of people. So, of course, I'd like to thank Dr. Dave. And he looked over this and he's like, why is this picture up here? Well, this is a picture of one of the first days I started my first independent study with him and with the viruses. And I mean, I can't say enough. It's without you, I probably wouldn't have the job that I have after college, and I wouldn't have the opportunities, and thank you for all the knowledge and the continuous knowledge, so I am forever in debt. I would like to thank someone who doesn't get enough recognition all the time, and this is from freshman year, and this was at Lake Forest, which, if their pool is an actual dungeon, and this smiling individual kept me going the whole time, and he doesn't get enough credit, and I want to really acknowledge you, D-Mark, because without you, you've been there with me at the hospital, you've been there with me in the stock room, we've been jamming out to, Missy Elliott, and then obviously ordering all the supplies needed for this research. So, Den Mother, I can't thank you enough for all that you've done over the years. Thank you. I would like to thank the Lawrence University Chandler Senior Experience Fund, because without this money, I would not be able to have any of this, any of the research. You need funding to do things. So thank you very much, Lawrence. I'd like to thank Bethany Condas, because if it wasn't for her, I wouldn't have this research, because she's the one that started everything. So without her, I would be nothing. Not quite true, but I'd like to thank the LU Chemistry Department, Graham, Stefan, Allison, Deanna, Mike, D-Mart, everyone, because without you guys, I found my home in the chemistry department, and that's hard for a hybrid like biochemist, because you can either go one way or the other, so I really appreciate that. All of our blood donors, because Lord knows we would not be able to do the research we have without you, so thank you all. I cannot say anything, because I'm under, consent forms and whatnot, so I can't say your name. Thank you. And then also my family, my friends, thank you, Mom, thanks, Grandma. Thank you, all my friends, for coming out. It really means a lot to me. And specifically, I'd like to acknowledge these gems. I had to give some really nice pictures, but without them this year, I would be nothing. They supported me through everything, and I would like to point out right here, this ride or die. She is my roommate. She's a real gem. Anyway, I'd like to thank all of them for all their support. And any questions?
This is your chance to talk science with Shelby. Come on, ask questions.
I was going to be really sad, Gus, if you didn't say something. I really was.
I gave everyone else the opportunity to.
Okay, thank you.
So looking at the TNF plot where you have the picograms per mil,
Yeah, hold on. Wait, the TNF alpha one, the
Yeah.
Bethany?
right, so yeah, so that expression level, is it like 3,000 picograms per mil? But
Yes.
you're looking at,
Approximately, yes. Sorry, it's hard with, I should have
But
put
then
some.
looking at interferon beta, you said it was more like 10.
Yeah.
Does it just have a much more, is it much more effective at lower concentration, or like what's the expression?
I mean, yes, it is. Also, TNF alpha, and that was also lipid polysaccharides, which it's going to be different expression between viral and pathogen bacteria. It's going to be a different type of stimulus produced. So that's essentially what it is. Michael.
Do you have any ideas about the differences in the upregulation of interferon beta between the GABA treatment and the GABA with human rhinovirus?
Could you repeat that just one more time?
Well, there's a decrease with the GABA with
Yeah.
all the way to number 4, and
Yeah.
then number 2 with just GABA, there's
Yes.
way more regulation. So I'm wondering if there's any ideas about why there's a difference there.
Because the mechanism, not entirely sure. We're working on that. However, as you can see, the stimulation is through the roof with GABA. So the whole point of this is that it does have inflammatory mediation properties, which is really cool. And essentially, most people think that it inhibits and just pumps the brakes, but this obviously ramps it up, which is really, really cool. And I guess you're not going to see the same amount when you have virus. So it's kind of like, this minus this equals this, kind of, not quite, but that's kind of the idea behind it, is that GABA will still amp it up in the presence of a virus. So the mechanism, though, we're working on, but that's the gist of it.
More questions? Okay, let's thank our speaker. Oh, hold, there's a question. Oh, I missed one, sorry. Go for it, Lynn.
So in the first lecture, you said GABA is kind of close the break on the
Yes.
inflammatory,
Yes.
but then here it increases the IFN caffeine.
Yes.
So is that how
Yeah.
how
So,
they would show?
yeah. So that's essentially that's why when we phrased it, it was mediation, because originally in the central nervous system, actually GABA has a really important role with when you consume alcohol and drink alcohol and consume barbiturates or take barbiturates. It's the same. They have a inhibitory, specifically in the central nervous system. However, in the immune system, this seems to not be the case. It can amp it up or dampen it, so it's able to mediate. It's essentially the middleman on how this is expressed. So.
Good questions? Other questions? Your last chance. Okay, let's thank our speaker again.